Hi everyone and welcome back to my channel. In today's video, I will explain you step by step how to perform identification analytical method validation as per ICH guideline with one example. So let's start it. For reference, I use paracetamol monogram for identification AMV. In paracetamol monogram, there is a one taste IR by identification. Let's start it. I use blank AMB protocol for identification by IR. In first page, we have to mention protocol product name. It's a paracetamol and it's a BP. Then taste identification by FTIR and you have to mention what model you use. It's ATR model. Then we have to mention report number, manufacturer name. In second page, there is a table. We have to mention all the parameters in this table. In third page, it's a approval for analytical method validation protocol, sign date. And fourth page, it's an abbreviation. I mention all the abbreviation here. Then it's a propose. The purpose of this protocol is to verify the validation analytical method for determination of IR of paracetamol by FTIR spectrometer as per BP. Because we use British Pharmacopoeia for paracetamol. Scope. This protocol covers the method of IR of paracetamol by FTIR spectrometer. Responsibility. QC chemist officer to analyze the sample as per approved analytical method validation protocol and record the result. QC executive and designee to prepare validation protocol verification report and review the analytical validation to ensure that the approved method is followed. QA/QC manager head to approve the analytical method validation protocol and verification report to ensure the method validation is done as per procedure and after verification test method followed. Then we have to go next page. It's a reference current ICH Q2 R1 guidelines. Then method validation procedure. Instrument is calibrated, observation yes or no and was checked by signed it. Analytical chemists are qualified. If chemist is qualified then yes and checked by. Then detail of the method to be verified means name of the test method, the determination of IR, paracetamol. Then pharmacopoeia or non-pharmacopoeia. We have pharmacopoeia method. It's a BP. We have to mention that. Then test parameter to be validated as per ICH. Reference. It's BP. Product used for validation. It's paracetamol. And reported by. Then following analytical method parameter are to be carried out. It's a two parameter, specificity and precision. In precision, we have to do method precision. Method description. The details of the methods given below. So, principle IR of paracetamol is done by FTIR spectrometer as per BP. Sample needed. The following samples are used for analytical method validation of IR of paracetamol as per BP. Reference standard. We have to mention paracetamol reference standard. You also mention batch number here. Then paracetamol taste and also mention in bracket batch number. Then details, testing equipment, FTIR, ATR modules, IR conditions can range between 650 cm and 4000 cm. Procedures. Perform the background measurement with no sample being placed on the ATR crystal and no contact between pressure application device. For the background measurement, the measured medium is air. Then after background measurement, grind a powder sample if necessary. If there is no fine powder, then you have to grind the sample. Throughout using a mortar before putting it the ATR crystal. Place working standard on Zense crystal of FTIR and press the sample against the ATR crystal by using the integrated pressure application device. Run the spectra in range 650 cm to 4000 cm. As soon as the sample measurement is finished, release the pressure application device immediately and swing it out of the measurement position. After that, the ATR crystal has to be cleaned using a suitable solvent. This procedure is followed for the test sample. Compare the spectrum of sample with the obtained with working standard. And your acceptance criteria, IR absorption spectrum of the test sample should be concluded with the date of reference standard. In specificity parameter is the ability to assess unequivocally the analytic in the presence of component which may be expected to the present to prove specificity following experiment is carried out. Procedure as per 8.0 method description we mentioned already above. Perform the IR of paracetamol BP reference standard and sample by FTIR spectrometer in the range 650 cm to 4000 cm. Acceptance criteria. IR absorption spectrum of the test sample should be concordant with that of working standard. Here you can see the comparison of paracetamol working standard and paracetamol sample IR spectrometer. Here it's a paracetamol working standard graph and here paracetamol sample IR graph. Software compare that graph and purity index is 0.99968 means it complies. Now we have to go second parameter, it's a precision. The precision of an analytical procedure expresses the closeness of agreement between a series of measurements obtained from multiple sampling of the same homogeneous sample under the prescribed condition. Method precision. Method precision is determined by the perform 6 times analysis of same sample following the same method. Procedure as per above 8.0 method description. So in this parameter we have to run 6 different sample and then acceptance criteria IR absorption spectrum of the test sample should be concordant with the date of reference standard. So we have to run this type of 6 different sample of paracetamol and check their comparison and purity index. Then we have to mention conclusion in report. Then the validation. If there is any change in the manufacturing process parameter, the analytical method should be revalidated. Then records. The following records and reports should be readily available. Identification by IR spectrometer, analytical method validation protocol. Second, report. And third, identification by IR spectrometer, analytical method validation absorption spectrum. This is protocol. Report is same. Just we have to mention in report our results, comparison and attached our IR absorption spectrum and we have to done our analytical method validation. Subscribing to a membership on my channel, you will gain exclusive access to pharma related formats, including essential templates, guidelines, and documentation that can help streamline your work in the pharmaceutical industry. Whether you need SOPs, validation protocol, audit checklist, or regulatory documents, I provide valuable resources to support your professional needs. Join now and unlock premium content designed to enhance your knowledge and efficiency in pharma field. I hope you learned something from this video. If you like this video, then like, share and subscribe to my channel. I will see you in the next video. Bye bye.